And our next speakers, two speakers, are Jamal Shirley and Sharon Edmunds, who are going to speak to OneHealth approaches to Trichinellosis prevention with the Nunavut Trichinella detection program. So come on up. Jamal Shirley is the Director of Innovation and Research at the Nunavut Research Institute, and Sharon is a Senior Research Advisor at Nunavut Tungavik Incorporated. Thanks.
Yeah, Unusaku tamasi. Yeah, Jamal Shirley ngoyong Nunavut kawi sakto lagi kuningak tunga Nunavut sila to driving ilagayawiyong. And I'm very honored to be able to co-present today with Sharon Edmunds of Nunavut Tungavik Incorporated. Our talk today will be focused around a new, well, relatively new diagnostic program we've developed in Nunavut based on a model pioneered in Nunavik to detect a parasite that infects wildlife and can create an illness in people called trichinosis. So Sharon will start by providing some more information on this parasite, how it infects people and wildlife, and the health consequences of an infection with the focus on the Arctic. I'll then share some information on how our diagnostic program was created, the test method we use, some of our realities, which could also be considered challenges, some achievements, and then reflect on the OneHealth, how this particular program fits within the OneHealth model. Although learning from all of your wonderful words today has really struck to me how much more we need to do to really make this a OneHealth approach. So, Sharon?
Thank you, Jamal, and thank you, everyone. I've really appreciated sitting in the audience today and just listening to everybody's perspective. The elders talk, thank you, and thank you for sitting all day, too. I've been thinking about you guys. Like Jamal said, I'm with not NRI. I'm with Nunavut Tungavik Incorporated, the holder of the Nunavut Agreement. I've been with NRI now for about 14 years. Started out as a health policy analyst, but I've spent most of my time working in the research sphere. And yes, I'm here with our wonderful partner, Jamal, at the Nunavut Research Institute. All right, so the Trichinella parasite, it is a nematode, it's a parasitic roundworm. It has global distribution. It is a parasite of carnivores and omnivores in all terrestrial systems and some marine systems. It completes all stages of development inside a single host. There are 12 recognized species worldwide. All species infect humans, with the exception of Trichinella pseudospiralis. It causes the disease trichinosis or trichinellosis, and it's a result of being exposed to the parasite. Here you see the adult male. It is smaller than the female, actually. The female to your right, she can lay up to 1,500, she can birth up to 1,500 babies, so baby larvae. Below, on your left, you see the reactivated, or the larvae, once they've been reactivated, released from protein. And here, they're tightly coiled, they're just born, they're trying to stay warm. So, Trichinella nativa. It is distributed throughout the Arctic and subarctic. An important note, this particular species is freeze resistant. So freezing does not kill the parasite. It's been documented in very many different animals in the Arctic: grizzly bear, black bear, wolverine, fox, Arctic and red, wolf, dog, because of course some of these same animals are fed for dog food to different dog teams. In the marine system, polar bear, walrus, orca, and beluga. It's not known to infect bearded seal, harp seal, and ring seal in Nunavut. And I just wanted to make a quick note for orca. Our team of analysts, led by Jamal here, were the first, and team of Inuit analysts working in the lab discovered for the first time on a global scale the parasite in orca. So our little team here in Nunavut did discovery science, which is pretty cool. All right. So basically how a person becomes infected, it occurs if you eat infected tissue, meat tissue, that contains the larvae. Once it enters the small bowel, this protective nurse cell that you see down here gets released, and that's done through exposure to pepsin, enzymes, gastric acid, so the natural state of your stomach. Once they are released from the nurse cell, within 48 hours, they actually sexually mature and begin mating. They produce, like I said, a gravid female can produce hundreds of babies. These same baby nematodes then basically use the lymphatic system to migrate out towards your muscle tissues. Once they reach the muscle tissues, they use the cellular structure of your muscle tissue to actually create this nurse cell. And they basically go into a dormant state at that point. So, symptoms of trichinosis in humans. The severity of illness largely depends on the number of worms that are consumed or larvae. Symptoms can occur 2 days after eating infected meat. So this would be the early stage part of the infection. Symptoms can include nausea, diarrhea, vomiting, abdominal pain, and constipation. And very often, I'll note this, very often, these symptoms are actually, there's a lot of misdiagnosis that goes on with trichinosis because the symptoms could be the flu, it could be a cold. It's like, oh no, you have a stomach flu. But after 2 weeks, so this is in late stage infection, the symptoms worsen. Symptoms can include muscle pain, fever, swelling of the face, eyes, itchy skin or rash, cough, neurological kind of like symptoms, cardiac heart symptoms as well. In rare cases that can occur. Early diagnosis is super important. And I think that this particular point sets the stage for why our testing program is so important. The window period to treat a Trichinella or trichinosis infection in humans is 3 days. And it's an antiparasitic drug that has to be administered right away. So again, this is why our early testing program is so important, because it's a way of preventing exposure, potentially have to look at getting drug therapy into communities if there is an outbreak. So testing your meat first before consumption protects individual health, community health, and really continues to help Inuit promote food sovereignty and, that all-important traditional diet, which has so many benefits, which people have talked about today. Worms, once a person is infected, they will live in the intestine for up to 1 month. The adults eventually die, and they are released through bowel movements. And there are many reports of trichinosis in animals and people. Here you see Canada, you see Greenland. But because of the really close and important link that Inuit have with traditional foods, we tend to see outbreaks more so in the territory. And again, that is why we created this program 8 years ago. So back in 2007 and 2008, there was a very large Inuit health survey that took place under the International Polar Year. And in that study, there was a seroprevalence study done, like a sub-study, looking at antigens for infection. Trichinellosis was one of them. And in that study, we found that 16% of Nunavut Inuit they were seropositive for infection. Anyways, I am going to hand it over to Jamal.
Thank you, Sharon. So this, I would also add that the parasite is only infective in meat that is consumed raw or undercooked. Cooking the meat kills the parasite and renders that meat completely safe to eat. But as Sharon mentioned, because raw and fermented and frozen meats are such a vital part of traditional diet, the risk of this infection has been a concern for many years and prompted the Makivik Corporation to work with the Canada Food Inspection Agency to develop a special method to test samples from animals to determine whether those animals are infected with the parasite. And this program has run very successfully in Qaujigiartiit for many years, and I think beginning in the early 2000s, the Nunavut Department of Health started to send samples to Qaujigiartiit for testing from animals harvested in Nunavut after outbreaks here in our territory. This proved to be a little problematic because of the long delays it took to ship these samples over for testing. So our harvesters waited weeks sometimes to receive their test results. And this prompted the Nunavut Tungavik Incorporated to begin calls for the establishment of a testing facility in Nunavut to provide faster return of test results to Inuit. It was championed by the late Raymond Ningiochek for many years. And I'll be frank in that there was some difficulty in identifying a department within the government of Nunavut or another agency to take the lead in establishing that facility. However, the president of Arctic College in 2017, Joe Kunuk at the time, decided that we would take the lead and establish this facility using the model developed in Nunavik. And that's what we did. With the very strong support and guidance from Nunavik and from this Canada Food Inspection Agency, established a testing program here based in our lab facility at the Nunavut Research Institute in Iqaluit. Trained a team of analysts from the Nunavut Tungavik Incorporated and other local organizations to carry out this testing procedure. And we've operated continuously since then. So what we test at our lab are the tongues of primarily walrus, but we also test polar bear. And the tongue is called a predilection site. If an animal is infected with this parasite, the parasite are most likely to be found within certain muscle tissues of the tongue, or of the animal. And the tongue is one muscle group that is known to be a source of the parasite. So we receive walrus and polar bear tongues from throughout Nunavut, and we put them through what is called the double separatory funnel method. It's essentially an artificial digestion procedure that mimics what happens when an infected meat is consumed by an animal or person. So this involves blending a sample of the tongue in a mixture of pepsin and hydrochloric acid and water, then incubating that sample to effectively copy what is happening in the stomach of the consuming animal, and then pouring that mixture through sedimentation funnels where the larvae, if they're present, will sink to the bottom and be recovered on a petri dish and then identified under a microscope. It's very labor intensive, but something that our team of analysts who are students and graduates of our environmental technology program in the college are able to do incredibly effectively with great success in recovering larvae from infected samples. So to date, we've tested over 366 walrus, 66 polar bear from many communities throughout Nunavut. The number of tests we carry out varies year to year. We had ranging from a low of 18 when we started the program to upwards of 100 in 2023. It's a really important service for Nunavut, and one of the key purposes of establishing this program was to kind of develop that capability in Nunavut to conduct advanced scientific work in the lab that is typically conducted by facilities outside of our territory. One of the long-standing concerns we hear is that results of analyses and tests that are done on samples provided by Nunavut are never returned or take a long time to be returned to our residents. So building that local capacity and for more accountable, accessible science was one of our key objectives. That said, I want to go back to a point that Karsten raised earlier, is that building and maintaining trust in this and confidence in our program has been a challenge, and it's ultimately required hard work. We have to deal with lots of logistical challenges, shipping delays and lost cargo, samples arriving rotten and really unfit to test, cross-contamination of samples occasionally, not always, not often. The challenge of maintaining a laboratory in the Arctic, like developing our quality system, our standard operating procedures, undergoing annual proficiency evaluations with the Canada Food Inspection Agency, ensuring that our lab test results are relayed to the Department of Health, who are responsible for then communicating those results to the hunters. Health and safety training and plans and resources are very important. These tests require a lot of hydrochloric acid and other hazardous chemicals and a lot of very expensive glassware and equipment and instruments, all to ensure that these test results make it into the hands of our harvesters in a timely manner. And the dedication of our team is really incredible. If we receive walrus samples, for example, on a weekend, our analysts are willing to come in and run the tests just to ensure that that meat can be distributed by the harvester or the HTO that's requested the test. And I would note, too, that funding for continued operation is never completely certain for this program. NTI has provided all of the resources essentially for this program to function. And we need to further explore other funding streams to ensure this continues into the future. But like Sharon mentioned, we have had some really important successes. We're the first group to identify or detect the Trichinella and an orca in Arctic waters. That's important as more carnivore species make their way into Nunavut as our climate changes. You know, we'll get more terrestrial carnivores as well, black bear, grizzly bear, maybe even beaver and other animals that might end up on people's dinner plates. So it's useful to have this testing service available here in our territory. We have research opportunities with our archive store of infective tissue that our team is developing research questions around, looking at the freeze tolerance and how long these parasites survive in the tissue we have in our freezers. And interestingly enough, we recover live larvae from samples that we've stored frozen at -20 for 3 years. So it's a really good model, I think, to develop new surveillance capabilities for other parasites and pathogens that might be coming into in the north, into Nunavut, and that could pose a problem for people. I'll close with just this slide showing the complex, flexible connections that are needed just in this one small program that is kind of builds on this OneHealth concept that Inuit much more eloquently shared than I could ever. But here, just with this one little program, we have numerous groups involved that all have to share information and collaborate. We have hunters and their families. We have Department of Environment conservation officers that help with receiving and shipping samples. Fisheries and Oceans provides an individual analyst occasionally. Canada Food Inspection Agency is our source for proficiency assessments and updates to our protocols. The Department of Health is responsible for issuing advisories when we do detect Trichinella in a species. The college and NRI provide the laboratory space and oversee the operation of the program. NTI provides the analysts and the funding, all to support the interconnected relationship between Iqaluit and Inuit. I hope I haven't put everyone to sleep here at Akunyalo Kauba Apekotik Sakawit Apelinaxusi. Ati Joe.
So I'm from what I think you already answered what I wanted to ask, but aging doesn't kill like in the walrus when you're aging it, that process wouldn't kill the trichinosis.
We don't have, there's no clear evidence as to whether it does or not, but Sharon might have more to add. Yeah.
Hi, I am a former CHR, and we get literature from other organizations about parasites and stuff like that in our animals. But because you can't see it with the eye, like the naked eye, like is there Inuktitut terms on these parasites? Or are they in English only?
That's a really, really good question. I haven't found one consistent Inuktitut translation for Trichinella for this particular parasite, but I'll give one example. We received of the strength of Inuit knowledge related to wildlife health in this parasite from Iqaluit. We received a walrus tongue sample that was infected with Trichinella. We quickly relayed that information to the Department of Health, who reached out to the hunter and the conservation officer, and they said, oh, it was an elder who harvested the animal. Said he already knew it was infected, and he buried, sunk the carcass in the ocean. How on earth he knew that is fascinating, and we need to better understand those techniques for diagnosing animal health and determining whether an animal is sick with this parasite, which is invisible. Yeah.
This makes me itchy hearing this topic, and I want to know if there's been any studies or knowledge of how the parasite spreads to all these different types of animals, like you mentioned, marine and terrestrial. So is there some kind of any understanding of how they move?
It's the same way we get infected when we eat meat that we've harvested. It's through predation. So it's interspecies predation. Sometimes it could be polar bear to polar bear, like an attack, and they're exposed that way.
How they infect walrus is a little bit of a mystery. There are different hypotheses, and one is that clams that are the walrus's primary food source can actually filter the parasites out of a decaying carcass on the seafloor, and they are a temporary reservoir for those parasites, which are then eaten by a walrus. And then the other possible explanation I've heard is that some walrus could be carnivorous and scavenge carcasses as well. But yeah, there are, as Sharon mentioned, the pathways of infection are the same for animals and people.
I just wondered if there are any reports about whether it's rising in numbers of infections, whether it's decreasing. Do you have long enough data to know what the...
Sorry, could you just repeat the question for the interpreters?
I was just asking if there was any data on whether there was a rise in the number of infections, or if... That's it.
So I invite Jamal to jump in here too. I think that we're actually establishing prevalence right now for walrus specifically. Like I know, like Nunavut has a sense of their prevalence. They haven't recalculated in a while, but also just through conversation, because their testing program has been so successful. They've been around so long, right? They have a lot of uptake in testing. But I think we're still on that pathway where we're still trying to determine level of infection in the different walrus populations.
If we have an idea of what symptoms look like in humans, have you had studies on what symptoms look like in the animals?
So what I've read in the literature is that the animals experience no symptoms, but their cycle of infection is the same way as it would be in a human being. So this is not based on research. This is just based on my humble opinion. Based on what I know, I feel that animals do experience infection. And then also, like, as our work has progressed and we talk to more harvesters and we hear more traditional knowledge about walrus health specifically and the observations made about their physical appearance, I feel like they have to experience symptoms when they're infected.
Inutitu nililang agama ila hope nana ila tamakwa asivaktiin sivulivulliila haujimanersausimammata tamakkunangan uumajuqtamining tusaumagama uumajuqtun ammaisuugummata pilasuugummata haujigiaqhaasuugummataguuq tusaumagamale tingungigguuq taima haujinaqtut uumajuqtaujuq taima hupiqqauhatuinariaqarpan tamannali tusaumagakku nillerutigiyaqa аммалугууннерхингин тухулигуннаратик хупеққулиит таманнатусауматтаққара таиккуа сибулерилаортапун уллумилугалуа инуужуллу илангин хаужимажут наамманаарсимангиппата инна уумажортаужоқ пияуниужангиттор тусаумажунга таматуманган кисиани uvikkai tamakwa asivahattamimata nalunaiaqbiuqannerialiin haujisatsiaqattarujauluti ippigusutsiaqrujaulutin uumajoqtamining haujimagasuaqutiqarlutin hupiqqorhatuinariarhangikkaluarmangaa inuujugu tamaani ukiortartumiugujugu tamakkunangan nerhittinning nerigumallerhattarssutalu taimaigatta hano tamanna sila malitsugu pitaqasuuq haujimagatta nerigumatsautigissuugutsutalu huaqataaminiapinnik ubbalu tikiujjaaqataaraapinnik nutaaning nerigumasuugugatta neriliniugatta inuujugut nutaaning tamanna nillerutigikumakainartaqa nalunaq pallaimman tamakkua hupeqquliit uumajoqtaviniutillugit ilanginnut nalugijaungittuugaluan taima haujimajupaalunnik pitalipaalu tamaani ukiortartumi tamakkua uumajuit hupeqquharniujajut ileqqusinginnik uvaniittuhangituinnapallaajok tamakkua Nilligu tauranga tillugit hupiqulilluunniin qaujisimayun qaeqqujauhatauhataq pata ilinnersauajaqpallayugun tamakkunangan qaujimagumayattinni. Qujannamik.
Apeqqutik saqatuinnartangala ila suurlu mayumut nunamiutama uiartauguma qupiqortaarunnarivunga
Apequtiqtiartiava. Actually, you would have had to consume part of that animal that is attacking you. You can only be infected with this particular parasite by consuming meat from an infected animal. So unless somehow that happened during the attack, it would be unlikely for this parasite we're talking about.
I really like that the question from here, if there is a name for it in Inuktitut, in our mother tongue, Galaslisut, Greenlandic Inuit language, we have a word for it. We say qumak. Qumak. And we also have a lot of information that my friend Eka can tell you a little bit about.
Yeah. In Greenland, we have stopped to make samples and yeah, we cook it. Yeah, we cook it. We just know we have to cook it for 3 hours, and then we just stop.
Then we can eat it.
Simplify and, you know,
because it can't betale sig. It can't. It's too expensive to all these lab investigations and all that. So the requirements are cook it for 3 hours.
Well, not because it's expensive, but it's not worth the effort, because the only thing you can do is to cook it for 3 hours. Anyway.
Yeah, to kill the parasite. Yeah.
Qumak. Koyanami. Uqausaqatiginginnata ilanikku we don't speak the same dialect, but we have qumak too, but it's a different one. It's in the intestines. Qumak. Parasite, another kind of parasite, but I'm not sure what's it. It's long, those long worms. But it's a parasite. But these ones we're talking about is trichinosis, which we can't see. Koyanami.
This could possibly be the most discussion on Trichinella in a public forum in a long time, so this is really exciting. Does anyone have any other questions? Just one comment, I think, about the polar bear cooking for 3 hours. This is definitely how people enjoy polar bear meat in Nunavut, and definitely the recommendation for eating polar bear. And I think, just as an observation, one of the big differences is that people do enjoy walrus fermented and uncooked before it is fermented. So yeah, it's great to have these discussions. Any other questions for any of the speakers before we close? Oh, okay, 2. Lucy?
Qaujigiartuinnartunga apeqituinnartunga taimanna quperrisemayumi aannia semayumi aktorsitilluta aittortaujungnarivitaqai.
So you can't get infected by touching the animal. The only way you can get infected is if you eat the meat that contains the larvae, if the animal's infected. And that's the primary way that a person gets the illness.
I'll also add one important point that I forgot to mention earlier, that this particular parasite, Trichinella, is only found in muscle tissue. So it's not in the fat, it's not in connective tissue, not in the cartilage, not in tendons, not in bone, not in the hair. It's just in the muscle.
Pitsula, last question.
Ape kutiula ngitu o kakomatuinartungale tamane khauji sabbita khalutilogo kuvealoalause magama ania khannangituleri yiu qattarsi magamata aqaaga ugasani CHR ukul taimaittu khatika gilautaakka tappa illinniatitsigasuartuni taatsuma mitsaano inuttitu liqasuartugu nalunagalaxattalaorsimayu hupiqquulasuungugatta asinginnittau kisiani nirtuti niittu hupiqqu taanna maanna akaulaanguvallai taqqo tamaanile qauyisainnaxattaxuyiyunga nunavummi oqaqqauma Jamal pigganartorsiuqattartuapi uvallaiui ilaannikku Kinau ja pitjutigillugu an akaungiliorutiillu saqqeqattapallai taqqu kisiani akaunaaqullarittaqa IV-tau qaujisartaunginnariaqaqattangata IV-minertortitau IV-minertortiugatta tamaani igunaliangusuungullutillu taanna qaujisarpi quviagillarittaqa aulatitsiviugunnanga teema qanuingippa uuttuutigilugu aive igunarsiangugunnaqajanga ila igunarsianguqattangata igunartuqattartutillu amisu inui taanna qaujisarpi quviagisimallarikakku taanna uqauseriguma qaujaqa tau quyannami.
Okay, Martha, last question. Last word.
Ilaaninlerumalermi jungalitainna illulimmiqaujisarpi naammaginginakku aulatitsiviugunnaqaluartillagu qangatajukku kisiani taavangakingarning aulatitsi yoqaqattaqajanga aulatitseqattangitsiamaqittugulle kingarni amisullukaatsutik nerigumallersuti taimaisuugumata nunalillimaalli uumajorniatikkungitigut uumajuit hupiqqusarmangaata qaujisaqutimin piqaqujaqaluakka nakormik.
Going to make Martha. Oh, yes. I'm not very good at this moderating job, Quet. I'm going to get fired. It's okay. Go ahead.
I wish Malaya was here so she could hear this question, but she needs the washroom. This is, I think, for the elders either at the table or anyone in the room. And it's kind of related to what Malaya was asking about symptomology in the animals. Is there a traditional practice for hunting like walrus and polar bears where you have to watch for like a sickliness or sickly behavior in those animals? And maybe does that help identify walrus or polar bears that might have parasites or might have another disease? I don't know, so I wondered if someone in the room has that information.
Thank you. Would anyone like to comment?
I don't think that I'd be able to identify a predator of some kind. It looks like mostly meat-eating things get this parasite, and maybe if there's a severe case, it would be noticeable. But I know that you can always look at the liver to see if it's unusual or different from like if you are skinning and looking at things, and as that elder seemed to have looked at the liver, and I guess in this case he already learned to identify by looking at the tongue also, I guess. We don't harvest enough of the walrus for my group to know the answer for this, but I'm realizing now that any predator seems to be able to have that. Not that I think I would eat a wolf, but it is something that where's the wolf getting it from? So it's interesting that it's everywhere, and that cold temperature doesn't have an impact on it. I've heard about this for a long time, but I've never really known until this presentation of the things that we should really all know about trichinosis. And I think it's important that we appreciate what is there now for people to understand that if they get these symptoms on their body, you better get checked out. But I would think I know with Inuit, no matter which if it's caribou or whatever it is you're harvesting, if there's anything unusual, I know we're not supposed to eat them.
Thank you, everyone. And I think that speaks to something that needs to have more conversations like this. So thank you, everyone, for your attention, and thank you to the speakers, my friends and colleagues, for participating. And I guess we reconvene tomorrow at 9 in the morning. Okay. Coffee at 8:30. Thanks, everyone.